Okay. 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 So, um, my primarily role at UCLA is, I'll call a collaborative faculty biostatistician, and I have collaborations across all the health sciences disciplines, medicine, nursing, dentistry, public health, and in many of the non-health sciences as well. So I lead a few biostatistics consulting groups, and I'm currently a co-investigator on 13 NIH-funded projects. And a lot of what I spend my time on is helping people write grant applications. So probably maybe collaborate with about 50 grant applications every year. And so thinking about study design and trying to position a project is what I really enjoy doing and what I spend a lot of my time doing. And certainly one of the things that I've found most useful in this is serving on NIH study sections. So I definitely recommend all of you who are, want an academic career and seeking NIH funding, that serving on a study section is probably the most useful and informative thing you can ever do. Okay, so the first half of this is really going to be focused on sort of how to interact with a biostatistician, what kinds of interactions are positive and what interactions might be negative. And so the first question is, why contact a faculty biostatistician? And these are the 5 main categories of consultation requests that I get. The first is questions about study design, and questions about sample size evaluation, how to develop appropriate analytical protocols for a study, data collection and management, and data analysis. Usually we think about statistics primarily as that last component, data analysis. Let's run a t-test, let's use statistical software and do a linear regression. Typically I find that that's the smallest and easiest part of my job, and often is something that occurs at the end of a very long process. And thinking about that, that as a statistical collaborator, I'm collaborating on the entirety of the scientific project and involved with all of the components of the study design and the carrying out of the study. If you talk to statisticians, one of the things they'll always bemoan is the lack of early contact with their collaborators. And it's my opinion that early contact between a statistical collaborator and their research collaborators is one of the key factors in influencing the success of a research project. So why do people keep saying contact the statistician early? What difference does it actually make? I'll start with the first one, and that one of the first elements, and you can translate this to not just statistics, but other quantitative collaborators and other members of your research team, that bidirectional knowledge exchange is extremely important. That as a statistician, I'll know statistical terminology, but I won't necessarily know the details of the research project. If somebody's coming to talk to me about COPD, I may not know what are the important clinical indicators. I may not know what the sort of epidemiology is behind the disease. I may not know what appropriate outcome measures are within the disease or what the clinical course is. But over the course of a collaboration, I can learn all of those things, and that will enable me to assist an investigator better if I really understand the clinical context of a question being asked. And ideally, I'll also be able to provide knowledge back, which is things like, how might we properly design the study, what might be appropriate analytical methods, things like that. Why else is early contact important? I think one of the things that I think comes out is this process of iterative refinement, that an investigator comes to me with an idea. I might have some initial thoughts about that idea, but over the course of meeting every week for a course of a month or 2 months, we can really as we learn, we can both refine our ideas and sort of come together on a plan that makes a lot of sense. What are other things? Identification of outcome measures. So often you might start a study with one idea of an outcome measure and have to change it because of feasibility. This is something that in group one we were just talking about this morning of in asthma, if the clinical outcome that was seen most interest was in rehospitalization, but potentially as an outcome measure that may not be all that great because it's potentially only going to happen to a small number of subjects. The outcome variable may not be very informative, and choosing a different outcome may make the study feasible, may make it so that we actually have sufficient statistical power to actually undertake the study. Other things are the overall design of the study, whether it's a randomized control trial, a case control, a cohort, any other design you want to mention. Often the consultations are based around the study population. What is the specific clinical context of the study? If we're going to be doing evaluations, what's that specific cohort, that specific clinical question that we're going to be trying to answer, and making sure that our outcome measures and our design are really appropriate in that specific scenario. So that matching is something that doesn't necessarily come immediately because you need to know enough about the context to really know how to do that matching properly. Another thing that's very important at the beginning is sort of envisioning the data structures, is imagining what the data is going to look like, and then, of course, what we're going to do with it at the end of it. So I always sort of make small doodles to try and sort of think about what the data will actually look like, what sort of the structure will be, and then think about how we might actually analyze those data structures. Planning data collection is another important area of collaboration, where what measures we'll collect, when will we collect them, how will we collect them. Let's skip the last one. So, what are some first contact with a biostatistician scenarios to avoid? My favorite one is the Friday afternoon email saying my grant's due on Monday, and can you please help? And the answer is probably not. Even if I'm feeling particularly nice that day, the likelihood that I could contribute something intellectually meaningful on a Friday afternoon is probably pretty limited. And without really understanding the basics of the research project, there's no way to make that really work in a sensible way. Another contact scenario to avoid is you have a study up and going, or the study's completed and a data set shows up on my doorstep. Now, that's many times the way it is, but if there's any challenges, once you've already gone into data collection, it's very hard to fix things that are problems. So, the third scenario is, and this is how many people contact me the first time, is they submit a grant application, the grant application is unscored, and there are a full set of statistical critiques on it, and they want those fixed. Now, at that point, I certainly can help and do help many people, but the ideal scenario is not to waste 9 months of your life on writing a grant application that's going to end up getting unscored. Sort of going and being proactive about it is much more useful than trying to fix a problem that exists. The other thing I find with, it's also very hard to rescue an application that's already had a bad score to begin with, because applications that have large weaknesses, the reviewers will only focus on those large weaknesses. And even if you fix those, there will still be potentially some moderate weaknesses, which won't have been mentioned because they were considered lesser issues, but may still keep a grant from being funded. The other component is the idea of sort of technical versus scientific collaboration. So when you interact with quantitative collaborators, it's important to think of them as a scientific collaborator, not just somebody who's going to run something for you. Running analysis is certainly part of it, but as I said, without the full surrounding knowledge and making sure to set up the research so that the analysis will be actually informative at the end is the key component. So what's I said, come talk to a quantitative collaborator early, when is a good time for the initial contact? And I usually think of this as at the time you have the basics of an idea, but it's not very well fleshed out yet. You have some ideas of what you want to do, some innovative plans, but you don't necessarily know how it all works out. In academics, we're not really trained to go and talk to people about things we're not really sure of yet. And the usual scenario is waiting until you feel like you have your ducks in a row before you go talk to somebody. But if you really want to make use of a sort of a collaborative research environment, getting input early on so that you can optimize your research proposal over the course of some months is much stronger. The next component I'd say is embrace the value of specialization, and there's that if you're a lead investigator, you're a PI in a grant, you're going to have lots of collaborators. You can't do all of their jobs, and if you tried to, you wouldn't succeed. And so the idea of make the best use of your collaborators and their level of expertise, and don't treat it as a failing that you're not necessarily able to do everything all by yourself. The reason humanity has gotten as far as we have is the increased level of specialization throughout history. And I think that team science is really the key to success in most of the research projects that I've been involved in. So grant applications, not just power calculations. The components that I work with my collaborators are very heavily on study design, analysis protocols. Often I assist people at just trying to put together specific aims, and sometimes we go through 5 or 10 iterations of a set of specific aims before it's really solid. Bold and red, most of the time people say, well, I just need a power calculation, tell me what the right number is. And that's exactly the wrong approach to take, because that only can become possible at the end of a long process of really understanding what the right thing to do is. One thing I often do a lot of also is budgeting and identifying appropriate statistical collaborators. Just as in any other specialty, there's a lot of different statistician specialties, and finding somebody who is really right and who knows either your content area or the specific types of methods that you'll be using is much more likely to lead to success, because they'll know what the pitfalls are in your particular research area. One thing I see many times at my study section is the study section complaining that the quantitative investigators have insufficient effort to carry out the proposed set of analyses. And so thinking through that properly and realizing it's not just running the t-test at the end, but it's helping setting up the database, thinking about the design, troubleshooting the design as it goes along, coming up with analytical plans. So there is a larger scale involvement that is really essential. Speaking about power analysis, I felt I had to at least say something since I've emphasized it. So when you are at the power analysis stage and trying to figure out what the right sample size is for your study, what are the things that you need to think about and be ready to discuss for that kind of consultation? The first is based on what's the study design and what is the primary study outcome. Without those two, it's impossible to even start considering what the right sample size for a study might be. Once we have established what the primary outcome is and how it will be measured and when it will be measured, it's important to have some information on what that data will look like. And you can get that information potentially from the literature, you can potentially get that from pilot data, and maybe neither one of those exists, but then it's time for good guesses. What sounds reasonable, what's not reasonable in the clinical scenario that's actually at hand. Many times people ask me, well, what should the sample size be? What and I say, well, what's the size of the effect? And they'll say, oh, I don't know. You tell me what that is. And I say, well, I'm the least likely person to know what the likely magnitude of the effect of your particular intervention will be, and you are the most likely. So it's a it's a collaborative enterprise, but it's also it needs both sides to really succeed. Now the next thing I spend a lot of my time working on is helping people think through their data collection and management for their studies. So thinking through what's for data collection, what variables are we going to be measuring, at what time points are we going to be measuring them, and in what form. What's the structure of those variables? Without knowing these critical details, we can never do, we won't know how to properly do the data analysis, and we don't really know how we should set up our data collection instruments. I spend a lot of my time helping people with study database design. So partially this is a defense mechanism for myself, because maybe about half the people who show up in my office have an Excel file with their data. And that's an immediate indication that something has probably gone wrong. Using Microsoft Excel, while it's a nice, useful spreadsheet, is not a scientific data collection instrument. It has countless features that will confound you and make it virtually impossible to collect and maintain high-quality, consistent data. There's lots of database solutions out there. The National CTSA program has developed a software package called REDCap, which has been implemented at a lot of academic medical institutions. It has its limitations, but it does really enable you to do consistent data collection in a way that Excel can never do. Other things that are important, I think, is data monitoring during a study. So you don't want to find out about your data problems at the end. If your study sites have a difference of opinion about how to interpret the study protocol and are collecting the data in different ways, that's something you want to find out at the beginning rather than the end, before you realize that your data is unanalyzable because of differences in how the data was collected. Certainly the data cleaning at study completion is a key component of what my group does. So what are some of the failure points that I will typically see in terms of data collection? There's this first point is collecting either too much or too little data. So with mHealth, with high throughput molecular data, we often collect huge data sets. And sometimes that can be a good thing, but sometimes we're collecting so much data that we don't know what to do with it. And we can spend so much time carefully collecting and cleaning a large database, much of which will never end up being used. So, while you don't want to leave things out, keep in mind your objectives and be focused on your objectives when deciding what you're actually going to collect. Of course, collecting too little can also be a challenge. If you're doing a study on lung cancer, you probably want to collect smoking data on your subjects, and you want to make sure you're tracking your subjects properly if it's a pre-post design, keeping track of who's who. But deciding what and how much to collect is not a simple task, and often involves a lot of iterative evaluation. One of the things that I think helps me trying to sort of operationalize this for people is that I'm thinking through the data analysis at the same time as people are showing me what their case report forms or their data collection instruments are, and then trying to say, well, this piece of data, what will that be used for? How will this inform our conclusions at the end of the study? And thinking through the rationale for each thing rather than the idea, well, of course we have to collect that, so we'll collect it, without an idea of what it will actually be used for. Missing data can be one of the most challenging issues when you're doing clinical data collection, and keeping that as part of your plan, and also handling it properly in your databases. The tradition of I've seen many databases where missing data is coded as 99, and then you're wondering why there's a lot of 99-year-old subjects in your data set. So if you're doing something, if you're setting up a database, set up missing data in a way that won't confuse you at the end of your study. And I think I've talked to a couple of those things. Consistency in data collection is also fairly key. Statistical software, data analytics are fundamentally dumb software packages. They will do exactly what you tell them, exactly how you're programmed, and they won't think through. They won't know that if you have a field called gender and you have some subjects coded as little m and subjects coded as capital M, those are different things for statistical software. And while this is a trivially fixable problem, if you have 100 or 200 such variables, going back and fixing them all later is a very substantial challenge, and you will forget something, I can guarantee it. If you have a good statistician working with you, they will forget something, I can guarantee it. So setting it up ahead of time so that you can avoid those problems by having logic and range checks, data consistency checks for entry, you can avoid a lot of those things down the road. This last comment, sort of data versus information. When people are collecting data, they're often sort of stream of consciousness putting that into a database without the idea that it's going to be used for analysis. So things like comments and question marks in a data field are only things that are going to make it incredibly complicated for you at the end. In terms of data analysis, I think the key point of this whole slide is look at your data. A lot of people come to my office and want to talk about their data, and they bring up the obligatory Excel file, and I spend time going through it, and I tell them a lot of things about their data, and they're very surprised that I know all this about their data. And they're like, well, how did you do that? How did you know? And I said, well, I looked through your file. I scrolled up and down, I looked at each variable, I used this slider bar, and I moved up and down. You'd be surprised by how little, once the data is in a database, it takes on this forest effect for a lot of people, and as scientists trying to see it as a database as trees rather than the forest, and to just really pay attention to your variables, do some basic summarization, you'll learn a lot, and a lot of the potential problems you can avoid by just carefully looking at it. Okay, sort of the last point is in terms of data analysis. I want to focus on the idea that data analysis is a very iterative process. The first iterative process is really between the researcher and the quantitative collaborator. It's understanding what is in the data, what's the objective, and making sure that fixing data quality issues. That doesn't happen in one meeting. That doesn't even happen in too often. It takes some time to really focus in on both sides really understanding what's there. Iterative collaboration on data analysis. When we start a project, we may have one idea of how the analysis should go. As the results come in, as we understand the data, those plans often will change and need to change because often ideas that we started out with turn out to be incorrect. Okay. I want to just go briefly to the case study that I have, and this is a collaboration that I've had with Vivek Shetty on the development of a point-of-care salivary biosensor for predicting PTSD. And just want to use this as a little bit of an example of a sort of a process that really worked. And the overall study design was trauma patients and at-risk population. We used what's called a probe design, where we prospectively enrolled the subjects for whom their outcomes would only be known at the end of the study. We had a biomarker development process. We built models. We plan to build models, and those models will be locked down and evaluated on a specific validation group, but that is from the exact same clinical scenario for which the model is being developed. Maybe I'll just take for the last minute, just focus on sort of what were some of the components of the grant development and conceptualization process, which I think made a really big difference. One, I think the first is that it was highly collaborative and iterative. We spent months discussing what the design should be, going into details about, should it be a test training design? How should we do validation? What should be the outcome variables? What are all the predictor variables that we're going to use? We had a long back and forth process where we would each make contributions to the approach section, sort of read them over. My sections of the grant evolved substantially over the course of the study, and as did the approach. And the key components of sort of study design and outcome measures needed to take quite a bit of time to do, and that had to happen with enough lead time so that the consequences of all those decisions could be crafted throughout the rest of the application. And that some things like developing the data analytic plans, those things can often happen at the very end, when after everything is completed, the analysis sections can be written, but only by really spending a lot of time on the study design together were we able to have enough time to really make that cohesive throughout the entire application. Okay. And it's my contact information, and I'd be happy to take questions.